How about now? Okay, good. We got that part out of the way. All right, guys. Let me get this thing closed. So I'm recording it, and I'll post this on the not the shared folder, on my YouTube channel. But first off, what's the questions? Somebody give me a question. You can type it or you can say it.
Can we go over the Krebs cycle again?
The Krebs cycle?
Yes.
Sure. Okay. Let's look. See. Nope, it's not far enough. Okay. Okay. Let me share this with you guys so you can see it. Okay, we should be good now. Okay, as far as the Krebs cycle is just about taking a 6-carbon molecule of glucose. Well, I'm starting with glycolysis, splitting it into threes, and to start with, you're going to put a phosphate on each one of those threes. And that's where we're going to use 2 ATPs up. So that's where we spend 2 ATPs and we're investing it. And then in a little bit, we're going to put another phosphate on here on the other end. And we're going to take these and swipe some of those hydrogens, and we're going to make out 4 ATPs because you got 4 phosphates. So we split 2 and we get 4, so we kind of net 2. Let's go down a little bit. We're almost to the Krebs cycle. That's all glycolysis. This is pretty good. Let's go down. That's too far. Okay. That's it, but here's what we need. Okay, so at the end, we're going to wind up with pyruvate or pyruvic acid. It's the same thing. Now, what's going to happen is drawing a blank here, let me think. NAD is going to come on that 3-carbon. He's going to grab 2 hydrogens, and then he's going to be NADH plus another H. But we're not concerned about that right now. So when he does that, this guy's going to get rid of one of his carbons. So instead of having 3 carbons, now we have 2. Those 2 carbons are going to join with something called coenzyme A. We just abbreviate that, CoA. And when they do that, they're going to become acetyl-CoA. That's the beginning of the Krebs cycle. Let's move down a little bit. Okay, so there's our NAD grabbing these guys. There's the acetyl-CoA. Okay, so we got 2 carbons, and there's another guy of 4 carbons, and these guys are going to join together, and when they join together, CoA is going to go away. because we don't need them anymore. So we've actually taken that 2 carbon and made it a 6 carbon. It's going to be citric acid, not really important. It's going to do a little rearranging, which is also not important. What is important is NAD comes through, grabs another couple of hydrogens, and we lose one of those carbons. So now we're down to 5 carbons. We come down and coenzyme A is going to join again. NAD is coming and it's going to grab another hydrogens or a couple, and we're going to lose CO2. So those first 2 carbons that we started with right there are now gone, but we're not finished because some of their hydrogens are still available. So this is what we're going to do. So this 5 carbon is still, I'm sorry, this one's 4. We lost that last one. It's got coenzyme A. Then just think of it this way. ADP comes and you get an ATP. And that's what they call the substrate level phosphorylation. In other words, it's an enzyme that does it. When it gets finished, the same 4 carbon is going to lose its CoA, and then FAD finally shows up, and he grabs a couple of hydrogens and takes them away. Again, you're going to have a 4 carbon. It's going to change, but that's not really important for us. NAD comes and grabs the last 2 hydrogens and takes those away. And when it does that, this guy changes back to what it was to begin with. It's oxaloacetic acid or oxaloacetate. That's the Krebs cycle. Now, since we had 2, 3 carbons, we're going to run this guy's through, and then after they get through, this guy's coming through. So we're going to spin that Krebs cycle, twice for each molecule of glucose. Oh, excuse me. Okay, let's go ahead and go to the ETC. This is what it looks like with the structural formula. You can see that, and they're even showing you the hydrogens that are going to get grabbed and taken off. And it's changing, especially when it goes down to 4 right here. It's just going to do rearrangements, and we're going to grab more hydrogens. And that's really all we need to know about it. Here's the electron transport chain. Now, that NAD is going to come and it's going to drop off its 2 hydrogens. So what's going to happen, those hydrogens and their electrons are going to be separated. So there's the hydrogen. They're basically nothing but a proton. And there's the 2 electrons. Those electrons are used to pump hydrogen out. Remember, this is the mitochondria. This is actually between the outer and the inner membrane. That's where we're going to put all those hydrogens at. Once this guy has done that, he's going to pass it off to this next complex, which actually transfers it to the next guy who's going to pump hydrogen out. This is going to be the same thing. He's going to be transferred to the final one, number 4, and he's going to pump it out. And after that, these electrons we can't use anymore. They're too weak. It's kind of like if your battery goes down below a certain voltage in your car, it won't crank. It's not 0, but it still won't crank. There's not enough power there. So, as a matter of fact, you have to have a certain amount of voltage to even jumpstart it. In any case, those 2 electrons, we're going to grab a couple of hydrogens, put them back together, and then you've got 2 regular atoms of hydrogen. You're going to take an O2 and split it. The 2 H's and the 1 O going to come out, and it's just going to make water. That's the end of that. It's just water. Now, FAD doesn't start at the beginning, so it doesn't actually make as much ATP, but this guy is going to start here, so he's only going to go through 3 and 4 rather than 1. So it's no big deal. Okay, once we've got those electrons out, we let them come in through this guy. And when it comes in, it actually causes this guy to spin. And that spin is going to take that phosphate there in that ADP, and it's going to give us our ATP, which is what we run on. How's that, guys? Does that kind of explain it? That's another picture of the electron transport chain. And yet another picture. Oh, that's the whole thing of carbon metabolism. Taking it from glucose, which using the energy of the electrons to pump the hydrogens out and then let them back. That is it. I think that's about it for that. There's a picture of the mitochondria, and that's that inner membrane space that we push the hydrogens out. The Krebs cycle and the ETC are all going to take place inside the mitochondria. Glycolysis happens in the cell cytoplasm. Showing that. That's not really. So how are we doing, guys? Are you good, or do we need to? Let's unmute you. I can. I don't think I can. Or maybe. Maybe I did mute you. Oh. Can't hear anything out of you guys. I'm not sure if you're finished with this or not. Yeah, let's just see what's saying in the chat. I can't see the chat when I'm doing this. Okay, well, that's no big deal. You can type in your answers or your questions, rather. Well, I'm guessing you guys are good with this. Recording we want to keep. Need to just stop sharing. There are quite a few folks here now. Okay. Where is my stop sharing? I wish they'd stop changing this. No. None of that works. Ah. Well, just don't see. Okay, there we are. All right. Anabolic and catabolic catabolism, how they're used in your body. Sure. Anabolism is just putting things together. You take things like amino acids or like glucose and you chain them together. Nucleotides, it's all the same. It's exactly like you got a pile of Legos. When you put them together, that's anabolism. Now that takes energy. That's kind of what's going on with that. Catabolism is when you take your Legos after you're finished and pull them all apart. You're breaking things down. Now, when you do that, it releases energy. That's why it takes energy to take ADP and 1-phosphate and jam them together. But when that ATP releases that 1-phosphate, that gives us the power to run our body. That's what we run off of. Okay. How would I ask a test question about that? Well, I mean, this is, as far as that's concerned, probably be more likely to see that in relation to the absorptive and post-absorptive, or the fed and fasting state. You know, during the fed state, we know anabolism is what goes on. And like I told you, I know that's counterintuitive. Wait a minute, we're breaking this stuff down. Yeah, we're breaking that down, but inside the body, we're actually making things like glycogen and proteins and all that. Metabolism is what we do when we're in the fasting state, which is like in the middle of the night when you take these things and break them apart and release them so we can have glucose. You use energy to break things down in catabolism. No, ma'am. You actually get energy from breaking it. So it takes energy to put things together, but when you pull them apart, it releases energy. Okay, how are we doing, guys? Anyone else with another question, or do we still need to run this one down a little bit more? You can just type your question on the chat. Okay, good deal. Well, you are so welcome, ma'am. What else you guys have on your mind? I mean, I know we haven't finished the urinary chapter, and we'll do that Tuesday. Loop of Henle. Oh yeah. Well, we'll get into it a little bit, but we don't want to get too much in there because we haven't even done that in class yet, but we'll get it. Okay. Let me come down here and share this with you guys. And let's see. Oh, there's my little thing to stop sharing. They did something with it. Okay, let's go down, find our loop of Henle. Oh, this will be a good one to start on. Move this guy off to the side so I can see. Oh. Okay, here's the loop of Henle. Remember, the nephron, you got this big hollow capsule. You got blood vessels that come in there and they leave. The coming in is the afferent, the going out is the efferent. They push things out of there like glucose and waste and amino acids and sodium and all this stuff. So then it goes through the proximal convoluted tubule. Let's just call it PCT. And that's where a lot of the stuff that's good that needs to be brought back in will actually be transported out. It's going to be transported out because we're going to let sodium potassium pump do secondary active transport. We pump all that sodium out. We let sodium come in as long, come in as long as we can use that same energy to pump glucose and amino acids. So that's how we get that. Now, the loop of Henle, this guy is where we're going to establish zones of concentration. So down here is going to be like 1200. You know, it's kind of arbitrary. It doesn't change from 900 to 1200. It's kind of a continuous thing. You might have 600 up here. And up here is just the normal 300. And just like it shows on this picture, this is the way this stuff is coming in. This is what connects to the PCT. This thing is going to let water out, but it won't let the salt out. So if you remove water and you leave the salt there, it just stands to reason that it's going to get more salty. I mean, if you have a teaspoon of salt in a gallon of water, you leave the salt out, leave the salt there and take out a half a gallon, it's twice as concentrated. And that's what's going down here is when this stuff is leaving inside the tube, it's going to be at those concentration levels all the way down to the bottom where it reaches 1200. Okay, as it comes up this way, it's actually going to let salt out, but not water. So it actually goes down 900, 600, and up here 300, but it can go as low as 100 depending on how hydrated you are. So what we're saying is, because these guys are kind of at the same level, the water outside the loop is also going to be at that same level. Not only that, but this blood vessel, which is running this way, is going to do the same thing. It's going to pick up a lot of salt over here. And when it gets to the bottom of here, the blood vessel is going to be 1200. But as it goes up, it's starting to get some of this water and it's going to wind up leaving there at that same 300. That's what the loop of Henle does. It creates concentration zones. They call it gradients. So all of those are at that. And the reason we do that is because this guy, the collecting duct, if you get ADH and you put a hole right there, then water is going to leave the tube until it also gets to that same 1200. And if you put it up here instead, you may concentrate it to 600. This is the way we concentrate our urine when we're dehydrated. I mean, if you're drinking water all the time, it's no big deal, but sometimes, like when you're asleep, you can't drink water. To keep you from losing too much water and get into some serious trouble, we're going to have this concentration gradient that's going to be able to do it. Here you see it the same way. It's everything over here is 1200. Now, whether this goes out, it just depends on whether you put one of those aquaporins there, and that depends on ADH. That's the max we can concentrate our urine, and 100 is the most dilute urine that we can make. How's that, guys? They call this the counter current mechanism. The reason they call it counter current is because the blood is going this way and the urine is going that way. That's why they're opposite. That's why when this guy is getting less concentrated, this guy's getting more concentrated. This guy's getting more concentrated over here, but this guy taking the water that this guy let out is actually getting back to normal. How about that one, guys? What y'all think? That's the last slide on that one. We're going to talk about it again on Tuesday, guys, before the test. But it is good to get your head around it. And I tell you what you guys need to do. You need to go and find some of those animations. It really helps to see this thing actually in motion. You can describe this in words, but there's nothing like a video to really help you get a hang of it. Let's have some piece of straw in my shirt. Okay. Let's better look at, see if we got any comments. Well, let's not comment. Let's see if I got it here. Yeah, chat. Well, I asked about that on the test. Well, it's so important. It's almost inevitable that I'll ask about it. I don't know to what degree, but you just absolutely need to know this. You're going to see a lot of diabetics when you get out there on the job, and this is stuff on the kidney are going to be super important. You can't wait till you get to nursing school or wherever to learn this. You got to do it now because you're going to have way too much else to jam into your head in nursing school. How will I ask questions about vitamins and minerals? Well, oh, you want to know exactly how I'm going to ask about it? Oh, I don't know. I really hadn't thought about it. I mean, there's several ways. I haven't looked in the test bank to see what kind of questions. Things like on the descending loop of Henle, the descending loop of Henle is impermeable to what can't move through it? Salt. The ascending loop of Henle is impermeable to water, that kind of thing. As far as the countercurrent mechanism, like I say, we still got another PowerPoint to go on that, but it's that osmotic gradient that it's creating out there is so critical for our ability to concentrate. Or you'll remember we talked about this, that I mean, dehydration don't take a whole lot to get in trouble with that. This is so critical for renal function. Okay, let's see what this other question about the vitamins and minerals. Well, tell you what, let me stop this one and I'll go open that other one and see what we've got in there as far as how to ask about them. Let's see. Well, I guess I need to share this with you guys. Okay. Okay, there I am. Let's move down. Yeah, it's down here. First thing you definitely want to know is which ones are lipid soluble and which ones are water soluble. If you remember B and C are water soluble, then everything else is lipid soluble. Or you could do the way I remembered, I just did the ADEK. But what do we need vitamin K for? Blood clotting. We talked about that a long time ago. Vitamin E is necessary for the immune system, or not the immune system, the nervous system. Vitamin D for bones. Without vitamin D, you don't get calcium. Without calcium, you don't make strong bones. Vitamin A is necessary for our vision and several other things, but just the most common thing for that. Calcium, sodium, potassium. You guys have had AMP1. We got to have that for our heart and our muscles and our brain. So that's kind of some of these other guys. Well, chlorine always goes with sodium. That's the way we have it. Salt, sulfur, phosphorus. Yeah, you need phosphorus to go around with calcium to make the hydroxyapatite. Magnesium, very important cofactor. I actually had a patient in the ICU with hypomagnesia. It was kind of interesting. Symptoms of excess and deficiency, I wouldn't say so. That's really more of a clinical application. If I ask you do we actually need copper in our diet? Then I expect you to say yes, but in trace amounts. So know that some of these guys you need them, but you just need a tiny little bit. And you can't have too much because iron is a heavy metal and you can poison yourself with that. How about that? Does that kind of answer the question? Okay. What else, guys? B and C are water soluble. A, D, E, K are the ones that are lipid soluble. So they have to be carried just like the dietary lipids do. Okay, guys, what else we got on our minds? I'll go ahead and close this, and then we can open it back up. That's a good one. We hadn't gone there yet. We do need to know how we make it, but not in excruciating detail. Let me see if I pull this up. Get it on here. Well, I guess I could share this with you while I'm looking for it. Okay. Maybe it was on the other one. I didn't think so. I think it is on this one. Proton pump. It might have been on the other one, guys. Yeah, this one's. Let's see. This is two. Let me put it on the first one. Let's stop that guy. I would have sworn he was on the first one, but probably not. Well, he's not on the first one either. There's only one other one left. I know we've got it. Just gotta find it. Hmm. Well. It was on this one that I had it on. I just didn't see it. There it is. It was. I should have looked. Let me share this one with you and then we can go over this. It's actually not that complicated. And we we have talked about this for a long time. Get my pencil. So how do we make hydrochloric acid in the stomach? Same way. We take carbon dioxide and water. We take that same old carbonic anhydrase, and we are not finished with him yet. We make carbonic acid. One of those is going to break off, and we pump that hydrogen ion out. What's left is a bicarb, and we actually push that back into the blood. Now, if I'm putting a negative ion in there, I got to bring a negative ion in. So I bring chlorine and put it out there. Well, you got HCl, that's hydrochloric acid. It's just that simple, seriously. Well, I mean, for our purposes anyway. And since we're putting a plus in there, we're going to take the potassium out of there and bring it in. That's it. The alkaline tide is what they call it when the bicarb gets pushed out. That's that same old chemical formula. And we're going to see that again in big time in the next chapter after the urinary. But we ain't finished with it. We got to see it in the urinary too. This thing is all over the place. So yeah, it's a good idea to know that one. Does that answer your question, ma'am? Sir? I don't know. You're unverified. Well, you know who you are. I guess that maybe means yes. Okay, good. Okay, guys, what else can I help you with? You are so welcome. What's on your mind, my friends? Oof, gotta stop stooping over the desk. More questions, guys? Oh, okay. There's a great question. Submucosal, and myenteric plexus. Okay. I want to say that's on the same one. Let me look. I think it is. Actually, that may be up there. And actually, maybe on the other one. And look and see. Yeah, that's going to be on that first one when we talk. Yeah, we'll go into that as well. Okay. Okay, guys. So the myenteric plexus is going to basically, just like the name insinuates, myo is muscle. They're going to control the muscularis externa. The submucosal plexus is going to have a little bit with the muscularis mucosa, but it's also going to control the glands, especially those guys, the goblet cells that are going to be releasing the mucus to make sure everything goes in there well. Does that answer your question, ma'am? That's real. I mean, that's the function is really all I really want you to know about it. You are so welcome. Okay, let me go to absorptive. Yes, I think that's going to be the last one. Yeah, it is. Let me share this with you guys. We can go over this in a little bit more detail. Let's get away from the temperature. Wait a minute, is it at the beginning? No, that's temperature. Well. Maybe it was on this second one toward the end. I think it was. Oh well, we'll go find it. It's at the very tail end of our metabolism when we started. Yeah, that's where it was. Okay, let me share this with you guys, and then we can go over that. Okay, absorptive state is, think of it as the fed state. So when you eat, plus the next 4 hours or counting the time that you're eating, that's the time it takes you to process all of the food that's come in, all those nutrients, to get them digested and to get them into the blood vessels and off where they're going. And this one is what we were saying when we were asking about anabolism. Oops, get my drawing pencil. Anabolism is going to be the main thing that's happening during the fed state, and insulin is going to be the guy that's running this. We're going to use glucose because we just ate, and almost everybody, unless you're on some fad diet, is going to have some carbs in your dinners, breakfast and lunches and all that. So that's the fed state. It's just the food processing state. That's really the way you could think of that. Carbs, we're going to break it up. And once glucose is broken down, it's going to be, some of it's going into the individual cells courtesy of insulin. Some of it's going to be stored in the muscles and the liver for later on. Lipids are going to be put together and made into triglycerides or whatever lipid is necessary, cholesterol, and used. The amino acids are going to be going to be immediately made into proteins. And if they're not, they're going to be changed into fatty acids and stored. Here's the thing, we do not store proteins or amino acids. Well, we store proteins, but amino acids are not kept in the body in any kind of storage like we do for glucose and lipids. If we don't have an immediate need for them, we're going to turn it into triglycerides. This is really just a carryover from the GI tract. The glucose, some of it's going to the muscles, some of it, we just already said that. Proteins, the amino acids are made in there, and if they're not, then they're definitely going to wind up over here as triglyceride. Excess glucose, if you have more, once we top these guys off and we've got all that we need, the rest of them are going to be turned into triglycerides for storage. And that's about it for that. So think of the fed state is when we've got so much glucose that we're going to not only use it, we're going to top off all the cells, just like you would stop at a gas station and fill up your vehicle. And what's left is going to be stored because we don't waste these things. Okay. We talked about that when we did endocrine. There's not a whole lot. Insulin, protein synthesis, it's going to signal the liver to make glycogen. It's going to, top off all the tissue cells. Okay, let's go down. I mean, these other guys are involved, but insulin is the main guy that's doing that. The fasting state, after that, like 4 hours, then your GI tract is somewhat empty. And at this point, we're going to have to start running on our stored energy. Now, the glucose is the one in the liver that's going to be earmarked for the brain because the brain wants glucose. The rest of the organs can take those triglycerides and directly use those. The brain we can take those lipids and turn them into ketones for the brain, but then that causes a problem with acidosis. So that's the post-absorptive. You're going to be breaking down more than you're making. So it's definitely going to be catabolic. Glucogenolysis, breaking down the glucose. Gluconeogenesis, the brain, the liver will make glucose for the brain from non-carbs. So it can take proteins, can take lipids, it can turn them into glucose or ketones, whichever. Fats are going to be broken apart. The glycerols go into the to the glycolysis. Those triglycerides, if you guys remember, are going to undergo beta oxidation, and they're going to be cut into tubes that are going to be just perfect to feed into the Krebs cycle. Yeah, if you're in a starvation state and you're completely out of glucose, used up all your fat reserves, then we're going to be doing the proteins. We're going to use the muscles first and leave the collagen because, again, the collagen is what holds us together, literally. We're going to break down the glycogen. We're going to mobilize the lipids and have them be transported to the cells that need them, especially like the heart and the skeletal muscles. Glucagon is going to be the guy that's running this. Sympathetic nervous system, the epinephrine can also be involved in that. Glucagon is the main guy. Growth hormone has they want to have as much glucose available to use it to build new stuff. And of course, cortisol, chronic stress. As far as this is basically the opposite of everything that we saw on the other one. It's not really that complicated. So we're going between the fed and fasting state. I don't want to see that one again. This is just kind of, I don't expect you to memorize this, guys. This is just to help you remember what happens during each stage. What you don't want to do is try to memorize this stuff. What you want to do is understand it. If you understand what's going on, then you can apply it. It's so much easier than trying to memorize. Memorization just does not work in the lecture. Okay. And stop this. I'm going to save my artistry. Okay, guys. How would you ask questions about this on the test? About the fed and fasting state? Oh, goodness. Again, I haven't looked at the test bank. I mean, we could ask you which hormone runs each of those. We can ask you whether anabolism or catabolism is the main thing that's going on. We could ask about what happens in the liver during the absorptive state. And then you should be able to say, oh, well, we're making glycogen. I got glucose coming out my ears. What happens in the liver during the post-absorptive state? Glycogenolysis, I'm going to break down my glycogen, and gluconeogenesis, I'm going to make glucose from scratch for the brain. Does that kind of make sense, guys? Oh, you're welcome. How do proteins in the urine look like? You might see that as far as visual, but it's really not, that's really not something that we expect anybody to be able to do. Now, cloudy urine as being an indicator of infection, yeah, you can see that. As far as proteins in the urine, that's really going to be something on a urinalysis. We're going to get a lab for that. But one of the main causes is damage to the glomerulus. Those little podocytes with those little filtration slits, if they get damaged, then you're going to be able to have proteins coming out, and it's going to be really bad. The kidney does not need to deal with proteins. And some of them are actually toxic, like the myoglobulin from the muscle cells. Okay, you are so welcome. What else, guys? What else we got on our mind? Angiotensin 1 and 2. I think that's going to be the first kidney name. Let me share this with you, and I'll hunt down that specific page. It's pretty straightforward. Let's cortex, we're still in. Oh, here it is. Okay. So here's the way things are running. If you're not getting enough blood pressure in the kidney, so that a ferret vessel opens up as wide as it can, and you're still not getting enough blood pressure, that means the blood pressure in the body is too low. And the kidneys can't work without that. I mean, you're not going to push stuff out unless you got something to push it with. So what's going to happen? The kidneys are going to release renin. And remember, this is the renin with one N. Don't let them get you, especially on the final exam if they ask that. So renin is going to go to the liver. It's going to cause the liver to release angiotensinogen. Angiotensinogen is going to go through the lungs. That's in the blood, guys. It's going everywhere. And in the lungs, the ACE enzyme, angiotensin converting enzyme, is going to change it from angiotensinogen into angiotensin. It's going to change it from angiotensin 1 into angiotensin 2. So the liver, the angiotensinogen is going to be enzymatically converted into angiotensin at the liver. In the lungs, the ACE is going to turn it into angiotensin 2. Angiotensin 2 is going to cause the blood vessels to constrict, and it's also going to go to the cortex of the renal gland and cause this guy to release aldosterone. And aldosterone is going to the kidneys and cause them to absorb more water, in a manner of speaking. But since we're already on there and it's not really part of angiotensin, we might as well go ahead and talk about that. So this is where aldosterone, this is something that folks get confused. So make sure ADH works on the collecting ducts. That's where it does its work. Aldosterone works on the DCT. Now, ADH is going to cause these guys to make those aquaporin holes for water to leave and go back into the body. Aldosterone is going to cause the DCT to have more sodium and chlorine put back in here. And if you put the salt there, then water is naturally going to follow it. So that's the effects of angiotensin 2. Vasoconstriction and sodium chlorine reabsorption, which is going to cause water to follow them by osmosis. Did that help, ma'am? Went a little bit beyond that, but might as well be complete. A little bit. It is a lot. It is. Simplify it. Okay. R is going to go to the liver where it's going to be A1. And it's going to the lungs where it's going to become A2. And then aside from the vasoconstriction, it's going to the renal glands where it's going to be aldosterone. A lot of times when you got something like this, if you can just put it bare bones, it really makes it easier. That's why, like in the lab, I told you guys, just if you want to know how to do this with the biliary system, the best thing is just to make a stick model and try to keep all that unnecessary detail off. Okay, well, looks like we're good on that one. Did I ever share this with you guys? I thought I did. I didn't. Oh, well. No wonder it wasn't so good. Okay. Anywho, let me go back a little bit and let you guys see some of my reasoning. So in any case, here's where we're going. It's going from the kidney, liver, going to the lungs because the blood flows in all these places. We get what we want, which is immediately going to cause some action, and then it's going to the kidneys, to the adrenal glands. And that's just, again, that's just where all this stuff is. I'm so sorry. I should have shared that with you guys to start with. Okay. Definitely don't want to save my artwork. I'll have to redo those PowerPoints if I do that. Okay, guys, what else do we need to vote on the phone? Definitely got to stop punching over these days. Yeah, we don't really use those PowerPoints on Blackboard. You need to go to the shared folder that I have for us, and then we can, then you'll see my PowerPoints, which have a lot more on metabolism. Nephrons, well, kind of did, but can do it again. Let me share this with you guys and not make that same mistake of talking when you guys couldn't see. Okay, now we got it. Okay, I'm just going to put it right here. So the nephron, get my pen first. All right, guys, so here's what happens. This is a capsule. You got a blood vessel going in, it's an arterial. You got a blood vessel coming out, it's also an arterial. That's the afferent, which means the incoming. And the one that's going out is the efferent. Okay. Okay, in here, you've just got capillaries. These capillaries are kind of all bunched in together, but that capillary is called a glomerulus. That's called the Bowman's capsule. Together, they call it the renal corpuscle. Actually, there's another tube that comes out of here. This is the proximal convoluted tubule. I'm just going to make this one single line. And then it's going to go down into the nephron or the loop of Henle. You're going to have a distal convoluted tubule. And then this guy is going to empty into a collecting duct. Several nephrons are going to empty into that same collecting duct. So the blood pressure in there is going to push everything that's small enough and has the right charge or no charge out. All of that's going to be pushed out. In the PCT, proximal convoluted tubule, what we're going to do is we're going to get the stuff that's good and put it back out. So these guys are going to get the glucose and put it back out. It's going to take it out of here so it doesn't go off in the toilet. Amino acids, some of the ions that we need, we're going to get all of those out. And here's the way we're doing it. And this is going to follow your question on symport and antiport. We're going to use the sodium-potassium pump to secondary active transport to get this out. We're going to let sodium come in if we can get glucose to go out at the same time. It's just what we were talking about in the digestive system about how we move things into the cells. Yeah, the short answer to your question is, yeah, we're going to need to know symport, antiport. Symport just means they both go in the same direction. Antiport just means they go in different directions. Okay, so that's what's happening in here. We're going to try to get all the good stuff we can. Shouldn't have but maybe microscopic, tiny amounts of glucose that ever wind up in the bladder. Same thing with amino acids. Should never be any proteins, unless something's wrong. Now, this guy is where we set up that medullary gradient, that osmotic. Since we only let water come out this side, this is the way it's flowing in there, and then it's flowing up on this limb. Okay. So we only let water out over here. So as it goes down that tube, it's getting more and more salty. Now, when we come up this way on the ascending, we're letting sodium and chloride, and I think potassium too, come out. So as it goes up here, it's getting less and less salty. And this guy is actually creating, since the water inside here and the water out here is going to eventually come to a balance. If it's 1200 right there at the bottom, it's going to be 1200 right here outside. And there's a blood vessel, the vasa recta, that goes this way. Now, this guy is flowing down like this. This guy is flowing in the opposite direction. So as the blood vessel comes here down, since this guy's letting out salt, that salt is getting in the blood, and it's going to get saltier as it goes down. But as it comes up, it's getting less salty. It's going to actually go back to its normal 300 milliosmoles. The DCT is one of the places where we do the fine tuning. You can get rid of things like hydrogen ions if you're in acidosis. If you've got too much potassium, you can throw them out. We can let aldosterone bring in sodium, which is going to bring in water. And that's for the fine tuning. The collecting duct, ADH is going to work here, and it's going to put those little holes, and this hole is going to let water out until the fluid in here is the same level as all this. So if you call this 600, and if I put a pore right there, it's going to get to 600. And that's just the way we do that. We concentrate our urine. Okay. All right. Definitely don't want to say more work on that. Okay, guys, what else? What else we got on our mind? Whew. You are welcome back. Well, actually, I've got, for metabolism, I've got 3 PowerPoints. And again, you can look at those. And I've actually have videos on my YouTube channel of me going over those PowerPoints, one at a time. And that's also another good resource. So what I suggest is go to the shared folder, and get those PowerPoints, and then you can look at those and then go to the videos. And then you can shoot me an email. Oh, you've been doing that. Okay, cool. Well, just look at them all, and anything you don't understand, you can shoot me an email, or if any of you guys want to, if you can, come on Monday afternoons after I get finished with A&P1 lab, and I think it's at like 1:15. I'm there for you. I'll be glad to tutor. I'll be glad to answer questions, whatever you need. But we still got time left on here. If I do, I don't know if any of you guys have someplace to go, but just whatever your questions are, I'll do my best to answer them for you. Catabolic and anabolic pathways. This is, it's really almost like an AP1 kind of question. All of the anabolic pathways are going to be dehydration synthesis. You're going to pull an O from one of the molecules and OH from the other, make a water out of it. The O that's left is going to connect the two molecules. They have to have an enzyme. Catabolic is hydrolysis. You put the water back and it breaks it up. As far as specifics on this, about the only thing you need to know, you need to know beta oxidation cuts our guys up into twos. That glycolysis or the glycerol feeds into glycolysis. You need to know the amino acids feed in at different parts. Some of them will go straight into the Krebs cycle. Some of them will go into glycolysis. And remember, we talked about oxidative deamination and transamination. And all that is the amino acid would get rid of the amine group, which basically is going to be ammonia, and we give it to another guy, and eventually that guy's going to give it away in the liver, and we're going to combine that with CO2 and make urea. We've already talked about carb metabolism. Amino acids, we don't really talk about anabolism so much here. That's something we did in A&P1. And you're not going to have to go over that again. Nucleotides, not a whole lot. We did that in A&P1 too. And the lipids, just know lipolysis is when you break them down. Beta oxidation is a specific kind of lipolysis. Lipogenesis is what you have when you're in the fed state and you got too much of everything, so you just turn it into lipids. So yeah, it's not going to be a lot of heavy duty chemistry on that, guys. You just need to understand what's going on. Understanding is worth a ton of memorization. Let's see. Yeah, I can do one on metabolism. Let's see, what was the last one we did? Last one we did didn't work, did it? No, I'm sorry, that was A&P1. I can't remember which one. I think we did the ones on the GI tract. So yeah, digestive 3. So yeah, we could do that. What I'll try to do, how about I make one quiz, 20 questions over those 3 chapters, or 3 chapters, or 3 PowerPoints. And I could make you guys one over the renal system over both PowerPoints. I could combine those and make one. It may be like 30 questions, but it might be worthwhile. Yeah, I think we ought to do that too. I think that would be a good thing. So Monday, I'll get one of those or get that quiz made up, and we'll do it first thing. And then that way you can at least see what you're weak on and what you need to focus on. But yes, I will get those for you guys. You are so welcome. Anything else, guys, that I can help you with? Yeah, we've had a little bit of drop off. It's okay. Folks got stuff to do, I understand. GFR, net filtration pressure, and glomerular filtration rate. GFR is how much fluid you push through both kidneys in 1 minute. That's what GFR is. Net filtration pressure is, let me put something up here. At least I have something to draw on. And this time I've got to make sure that I remember to share. So you guys can see what I'm talking about. Okay, there we are. So the net filtration pressure, so I'm just going to draw a real simplified. This is glomerulus, just a capillary. Yeah, it's got those podocytes and those filtration slips. But basically, it's the same thing we talked about when we were doing the blood vessels. So here I've got 55 as my blood pressure. It's mmHg, but that doesn't matter. Now, I don't know exactly what I didn't memorize those off the PowerPoint, but let's say in here, since the proteins can't get out, they're going to pull back with 30 with osmotic pressure. So 55 minus 30 is going to leave 25, right? Now, if you guys remember, I said these guys were in a capsule. So when you're pushing fluid out and there's already fluid there, you're going to have a certain amount of pressure that's opposing it. In this case, it's going to be about 15. So you take that 25 and take 15 off of it, and there you go. And that's the same exact amount of pressure that we used in the when we were talking about the capillaries before. Only in this case, we don't have, on those capillaries, you have more pushing out, and then they zeroed out, and then you had more pulling in. It doesn't happen on this guy, but that's all net filtration pressure is. It's just you take all the pressures, and so right there, I've got basically 45 pulling or pushing, pulling, going back, 55 pushing out. So I got 10, and that's enough to push the stuff out. Okay. GFR, again, there are different GFR scales for different groups of people. And in the hospital, they will actually show that on the lab to tell you what's normal for any particular person in that group. The GFR is probably the main thing that we follow clinically on our patients. Now, we also do the creatinine clearance, but GFR is a big one. If you're not getting enough pushed out, then something is very, very wrong. You might have to go on hemodialysis. Okay, guys, any other questions I can help you with right now? Charts or diagrams? No, I don't think I've got any charts or diagrams that I usually put on this test. You are so welcome, ma'am. You so welcome. Hmm. Yes, ma'am, you sure can email me. You have a great rest of the weekend. Not much of it left, but have a great one anyway. My pleasure. Glad to help. Do that and just let me know if you got any questions. I'll be glad to try to fill in anything, any blanks that you might have. You have a great rest of the weekend too. I'll get this video posted of this review. All right. See you later.